Okay. Hello, can you hear me? Perfect. Hi, I'm Shivang Khandelwar, and today I would be talking about the lessons that we have learned from published literature of CTNNB1 syndrome. Around 3 years ago, I met Shpela with the help of Effie when I got to know that her son is affected from a rare neurodevelopmental disorder called CTNNB1 syndrome. Since then, I have been personally engaged with the foundation in finding a cure and treatment for the disorder. Together, we wrote a systematic review correlating the phenotypes and genotypes of CTNNB1 patients. Currently, I am a graduate student at Hebrew University of Jerusalem, where I am working on another neurodevelopmental disorder called PCDH19 cluster epilepsy under supervision of Professor Rotam Karni. So everything dates back to the year 2012 when this article was published in the New England Journal of Medicine. Here, the authors sequenced exome of 100 individuals and found the 3 novel mutations in CTNNB1 gene. And this marked the beginning of CTNNB1 syndrome, which was previously misdiagnosed as cerebral palsy. CTNNB1 gene is located on chromosome 3 at locus 3p22.1. It contains 16 exons, out of which exon 2 to exon 15 encodes for the protein. And the gene length is around 2300 base pairs. It encodes for the protein called beta-catenin, which is 781 amino acids long and contains 12 highly conserved armadillo repeats. A brief on CTNNB1 syndrome. It's a de novo loss of function mutation. What it means that it is not inherited. It is generally a novel mutation. And the mutation leads to haploinsufficiency, which means a reduced function. And that's why it is called de novo and a loss of function mutation. It is an autosomal dominant disorder, which means that for each gene we have 2 copies, which we get one from each of our parent. A mutation in either of it is sufficient enough to cause the phenotype. It is a rare disorder, as we all know, with the prevalence of 1 in 50,000 children worldwide, as reported by Chang in 2019. And the protein it encodes, beta-catenin, it is highly important in maturation and development of the brain. The characteristics include intellectual disability, global developmental delay, and speech impairment. So what is beta-catenin protein and its pathophysiology? Beta-catenin protein is highly involved in Wnt signaling pathway. But when Wnt signaling is off, beta-catenin is ubiquitinated by beta-trip, which is an E3 ubiquitination ligase, and it undergoes a proteasomal degradation. But when Wnt signaling is on, the degradation of beta-catenin is inhibited, and as a result, beta-catenin gets accumulated in the nucleus, where it acts as a co-transcription factor for many different target genes. So we know about the gene, we know about the protein. Now we need to know how the mutation in the gene affects at the protein level. One primary way is the mutation can lead to a premature termination codon. Due to this, the mRNA goes for degradation via the nonsense-mediated decay pathway. The cell decides that it's too much energy to invest in making a good protein, so it degrades it. And hence, there would be no protein. Another possibility is when the mutation would lead to an improper folding of the protein. Now, as the function of the protein highly depends on its conformation, a change in conformation would lead to a non-functional protein. Thirdly, it can lead to a formation of truncated proteins. And as the protein would be shorter, it would be a loss of partial function. Finally, though it's rare, but we cannot exclude the possibility of dominant negative effect, which means that there is a mutant allele and a wild type allele. So a protein from the mutant allele in certain cases can interact and interfere with the functions of the wild type allele. Currently, the open question in the field lies that if at all, is there any correlation between genotypes and phenotypes in CTNNB1 patients? And that's what we try to address in the review, correlation between phenotypes and genotypes of CTNNB1 patients. Here, we performed 2 novel analyses, including analyzing the clinical manifestations and doing the mutation classification based on the severity. For screening of the data, we used the following keyword, starting from the year 2012, because that's the year when the disease was first identified in different repositories, and we found a total of 1,221 articles. Out of these, we got the data for 84 patients, and we moved ahead with the analysis with those 84 patients. we found that the mutations for those patients can span across the whole gene, with arginine 535 stop being the most common mutation. Secondly, when we analyzed the data, like which type of mutation is most common, we found that nearly 50% of the cases showed a nonsense mutation, which is leading to a stop codon. Frame shift was seen in 35%, and missense and splice mutations were seen in less than 10% of the patients. And even 2 patients, which is 2.4%, was affected, had an entire gene deletion. There was no gene at all. On analyzing the data based on what exons are mutated, we saw that exon 9 and exon 10 serves as the 2 main hotspots for the CTNNB1 mutations. One possible explanation is that the exon 9 and exon 10 encodes for the region of the protein, which is highly important in interaction with other protein partners. So a mutation in such a crucial region is highly evident that might lead to CTNNB1 syndrome. So we next had to do a phenotype-genotype analysis. And in order to do that, we decided to segregate mutations and the patients based on their level of severity. To do that, we decided to choose 4 main characteristics which were shown in the patients. The first one is eye contact. For this, we classified the patients whether they were able to make an eye contact or not make an eye contact. For speech, we used the Viking speech scale, which was published in the year 2010. For cognition, we use the ID, and for motor development, we use the characteristics like whether they are able to sit and walk independently or dependently. It was so one thing to note in motor development that it was not analyzed for moderate and severe because they don't much differ between the 2 categories. So for example, if a person shows a positive eye contact with no speech and motor delays and a normal ID, was classified into normal. And for instance, a patient showing a positive eye contact with delayed and unclear sentences and was able sitting and walking independently with an ID between 40 to 70 was classified into mild moderate cases. And I would like to draw one more attention is like the patients who showed no eye contact with no speech and a severe ID, they were classified into severe category. On analyzing the data, we found that there is a specific set of phenotypes which was seen in more than 50% of the patients. And these we classified into primary criteria. This includes facial dysmorphism, eye abnormalities, muscle tone abnormalities, microencephaly, and speech impairment. Also, we found that there are specific phenotypes which were observed in less than 50% of the patients. That includes walking inability, aggression, stereotypic movements, autism, sleep problems, and intrauterine growth retardation. What IUGR means is that in the womb, the child doesn't look like it should. And so there is some abnormality in that case. One thing is of stupendous importance here for the physician is that if a patient shows at least 3 of these phenotypes listed in the primary criteria, they should be taken for a screening for CTNNB1 syndrome because it's highly likely they might have that. Another key finding was the missense and nonsense mutation towards the end of the protein, which is C-terminal domain in exon 13, 14, and 15, showed mild phenotypes. One possible explanation for this observation is that these particular mutations, they are leading to a protein which resembles a lot of it as a wild-type protein because only a shorter part of it is mutated. Hence, it can work most of the functions but not all, so only mild phenotypes were seen. Talking about clinical implications, we saw that this ability of the patients to speech varied from being non-verbal to having no delays, with 41% of the patients speaking a few words and 17% speaking short sentences and 6.5% speaking full sentences but a bit delayed. When we looked at behavior abnormalities, we found that nearly 48% patients showed aggression, stereotypic movements, autism, and sleeping problems were seen in less than 25% of the patients, and symptoms like ADHD, temper tantrums, and schizophrenia were seen in less than 10% of the patients. Facial dysmorphism, one of the key traits of the CTNNB1 patients. So small alveolar nasi, which is this part of the nose, is smaller, was seen in 97% of the patients. Long or flat plethysm, this area is either long or flat, was seen in 93%. Long, thin upper lip vermilion, upper part of the lip is thinner, it was seen in 90%, and broad nasal tip was seen in 87% of the patients. Talking about eye abnormalities, we saw that strabismus, which is misaligned eyes, was seen in 53% of the patients. Fever, which is a condition where there is a vision loss due to problems with blood capillaries, was seen in 23%. Hyperopia, which is farsightedness, was seen in 14% of the patients. Astigmatism, which is the vision loss due to an error in the refractive index, was seen in 9% of the patients. Astropia is where both the eyes align towards inward, was seen in 5%. Myopia is short-sightedness, again in 5%. And optic atrophy, which is the death of optic nerve. So optic nerve is involved in sending the signals to the brain, sending the visual signals to the brain. So it was observed in 4%. In the end, I would like to say that the supplementary data contains the list of 84 patients which were reported between January 2012 to October 2021 with relevant information. Review provides a phenotype-genotype correlation for CTNMB1 patients, and this can act as a database for researchers, doctors, physicians, patient advocates, and parents to correlate between the mutation and the expected phenotype. But one thing I want you to note is that the review does not confirm the mutation based on the visual phenotype. If you want to confirm the mutation, you should take the patient to whole exome sequencing. Thank you.